Hello, today we'll be extracting the DNA of my cheek cells. Our materials include salt. I have iodized salt because I don't have non-iodized salt. Water, if you can see it, it's in the thermos still. So it's preferably hot. We also need dish soap, so I also have an open concentrated dish soap. And then we also need cold 70% isopropyl, so it's still in the fridge. We need measuring cups, some cups in here, and I have a beaker here. So this is where I'll prepare my extracting solution. This is where I'll put something so I can gargle it, and this is where the final place where the DNA will be found. First, we'll need to prepare the extracting solution or the precipitating solution where the DNA from our cells will be. The extracting solution is composed of water, sodium chloride, salt, and dish soap. So first we'll dissolve a tablespoon of salt to 2 cups hot water. So first let's put 2 cups of hot water, so this is 1 cup. That is exactly 2 cups. And we'll need to put 1 tablespoon of salt. So we have 1 tablespoon of salt to 2 cups hot water. So that's 1 tablespoon. Yep. So what the hot water ensures is that the salt will dissolve properly. So now we'll have to mix it until it dissolves. So now we're done stirring this thing, and there is not much undissolved salt left. So yeah, the solution is clear. So now we'll have to gargle 1/4 cup of this, and we'll gargle it for a whole minute so that my cheek cells will mix with the salt solution. So in here I have 1/2 cup because I can't find the 1/4 cup, but yeah, we'll fill that halfway, so that's already halfway. I won't drink straight from this, so I have this, and now I'll need to gargle this, so that's a 1/2 cup. I'll start to gargle. So now we have 1/4 cup of my cheek cells mixed with the salt solution. That was 1 minute of swirling the thing. That didn't taste so good. So yep, here we have that, and now we'll add 2 drops of dish soap to the solution. So I'm just hopefully this is 2 drops, because I don't know. So yeah, I think that's 2 drops already. And then I'll have to mix it, stir it very gently. So we all know that dish soap breaks down grease or fats. Cheek cells have cell membranes and nucleus membranes that are made of phospholipids or fats. So the dish soap will break that down, causing the cells to lyse or burst. So now we can access the DNA, which is left untapped by the dish soap. Even though the soap is breaking down the membranes, we cannot see DNA right now because it is still dissolved in the water. And going back to why we added sodium chloride, the positively charged sodium ions dissolved in water will interact with the positively charged histone packing proteins that are associated with DNA, so it's further isolating DNA. So now we need to add a half cup of isopropyl, and this one is straight out from the refrigerator. So it says here 1/2 cup, so add a 1/2 cup here. This is 70% isopropyl. So now I'll have to tilt the beaker so that the alcohol sits on top. I spilled some, so that means I also have to add a few more. of isopropanol. Now, as I've said, DNA is soluble in water, but it isn't soluble in isopropanol. Since isopropanol is miscible in water, then what we're essentially doing is lowering the DNA solubility of the solution. When it has sufficiently lowered, DNA will precipitate. So we'll check in 3 to 4 minutes and see if a fibrous DNA will form. So it has been 10 minutes now, and if we check the solution, like this one, it formed right there. So there was a fibrous DNA. It's not much, but it is a precipitate. There may still be proteins in here, since salt may not be enough to completely interact with all of the proteins involved, but majority of this is DNA. So in here we can see how this thing floats from the top. And there's a separation between this is the soapy liquid, and this is alcohol, the isopropanol. So I'm able to get this thing. Look at that. That's not saliva. That's actually DNA. Ah, that's amazing. Right? That's amazing. Look, it's swirling around. That's my DNA.